Researchers just mapped the hidden wiring that keeps 20,000 brain cells ticking in unison, and what they found changes how we think about time itself. Here's what I mean. Your body doesn't just react to the sunrise, it predicts it. Deep inside your brain, a tiny cluster of neurons called the suprachiasmatic nucleus is running an internal clock that anticipates dawn before a single photon hits your retina. And it's not alone. Your liver has a clock. Your heart has a clock. Your gut, your skin, your immune cells, they all keep their own private time. And somehow, all of those clocks stay synchronized. Now here's where it gets strange. Take a completely different question. How does a zebrafish get its stripes? How does a sunflower know to open hundreds of tiny flowers in a perfect ring every morning? How does an embryo decide which side becomes the left and which becomes the right? These seem like unrelated puzzles, but new research is suggesting they share a hidden grammar, a logic of feedback, coupling, and selective stabilization that nature uses to build both when and where. Tonight, we're going to follow that grammar from the clocks inside your body to the stripes on a fish, from a sunflower's spiral to the handedness of your own heart. And somewhere along the way, we're going to ask a question that I've been sitting with for a long time, a question that runs underneath my own work on the Eden hypothesis. What if the move from unity to difference, from open possibility to committed form, isn't just something that happens to us? What if it's the deepest thing living systems actually do? I'm Chris. This is Jackdaw Patio. Pull up a chair. Lily, Charlie, and Dr. Vega are already arguing about it. Before we get into the deep weeds, let me set the table. Chapter 1 is about what's happening inside you right now, whether you know it or not. Every cell in your body is keeping time, not reacting to time, keeping it, building it from the inside out. And the story of how we figured that out starts with a plant, a dark room, and a French astronomer who couldn't leave well enough alone. Here's Lily.
Right, so picture this. It's 1729. A French astronomer named Jean-Jacques d'Ortus de Meyron has a mimosa plant on his windowsill. Every morning the leaves open. Every evening they close. Perfectly normal, yeah? But de Meyron does something quite wonderful. He puts the plant in a cupboard. Total darkness. No light cues at all. And the leaves keep opening and closing on schedule. The plant is keeping time without the sun. That's nearly 300 years ago, and honestly, I think we're still catching up to what that means. The plant wasn't reacting. It was anticipating. It had built something inside itself that modeled the rhythm of the world.
Hang on. A plant in a cupboard figured out what time it was? My phone can barely manage that when I cross a time zone.
That's exactly the point, love. The clock wasn't external. It was woven into the biology itself, and it took until 2017 for the Nobel Committee to properly honor the people who worked out how. Jeffrey Hall, Michael Rosbash, and Michael Young identified the genes and feedback loops that make this work. The period gene, the PER protein, this beautiful self-regulating oscillation where a protein accumulates, suppresses its own production, degrades, and then the cycle starts again. Roughly every 24 hours, every cell.
Actually, I want to slow down there, Lily, because that feedback loop is important. It's not a clock in the mechanical sense. There's no pendulum, no crystal oscillator. It's a delayed negative feedback loop. The protein builds up, walks into the nucleus, shuts off the gene that makes it, and then slowly falls apart so the gene can turn back on. The delay in that cycle is what creates the rhythm. That's the engine. And it's worth saying, mainstream biology is very comfortable with this. This is not speculative. This won a Nobel Prize.
So every cell in my body has one of these little feedback engines running.
Essentially, yes. Fibroblasts in your skin, cells in your liver, your pancreas, your gut. They all have their own intrinsic clocks. But here's where it gets architecturally interesting. They're not all freelancing. There's a conductor. The suprachiasmatic nucleus, or SCN, sits in your hypothalamus, receives light information directly from your retina, and coordinates the whole orchestra. But, and this is a crucial modern update, the SCN is not a dictator. It's more like a lead musician in a jazz ensemble. The peripheral clocks have their own rhythms. They can be influenced by food, exercise, temperature. The SCN provides a dominant beat, but the tissues are playing along, not just obeying.
I love that. One conductor, many players. And what's beautiful is that the conductor itself isn't just one voice either, is it?
No. Researchers at Washington University just published work mapping the functional connections inside the mouse SCN using a tool they call MITE, which stands for Mutual Information and Transfer Entropy. What they found is that out of roughly 20,000 neurons, only a small subset act as hub cells. Those hubs keep everything synchronized. Remove them or weaken their connections, and the whole system drifts.
So it's not just that you have a clock, it's that you have a network of clocks that have to stay in sync. And if they don't,
Then you get what researchers call circadian disruption. And the clinical picture is genuinely sobering. The American Heart Association released a scientific statement connecting circadian disruption to obesity, insulin resistance, hypertension, cardiovascular disease. A study published in Neurology found that people with weaker circadian rhythms had nearly 2.5 times the risk of developing dementia over a 3-year follow-up. Shift workers, people with chronic jet lag, irregular meal times, they're all living in a state of mild internal desynchrony. Now, quick caveat here, because I want to keep us honest. Correlation is not causation. In chronotherapy, the idea of timing treatments to the body clock is promising but not uniformly settled. A 2026 meta-analysis on blood pressure medications found no significant difference between bedtime and morning dosing for major outcomes. The broader literature still shows genuine timing effects in cardiovascular risk and heart failure progression. But the clinical translation is uneven. So timing matters. But timing alone isn't magic. We should say that clearly.
Fair enough. But here's what stays with me. Your body doesn't just float through time passively. It builds time from the inside. It creates a rhythmic structure, synchronizes it across tissues, and uses that structure to anticipate the world. That's not a clock. That's a kind of knowing.
all right, I'll give you that one. The body builds time. But what's that got to do with zebrafish stripes?
Oh, Charlie. Everything. But you'll have to wait for chapter 2.
She always does that.
Yes, she does. And somehow you keep falling for it, which is a pattern I find fascinating for entirely different reasons.
Chapter 2 takes us from time to space, from clocks to shapes, and it starts with a question that sounds almost philosophical. How does a field of nearly identical cells decide to become something with structure? How does sameness become difference? The person who first gave us a mathematical answer was a man better known for breaking codes than for biology. Here we go.
Alan Turing, 1952. 2 years before his death, he publishes a paper called The Chemical Basis of Morphogenesis. And in it, he asks one of the simplest, most profound questions in all of biology. How does a ball of identical cells become a leopard with spots? Or a zebra with stripes? Or a person with fingers?
Just to make sure I'm tracking, you're saying a perfectly uniform field. Everything looks the same. And then just because of how two chemicals move at different speeds, structure appears out of nowhere?
That's exactly what Turing showed, at least mathematically. And it's been confirmed in chemical systems. The SEMA reaction, a mix of chlorite, iodide, and malonic acid, produces Turing spots in a dish. No template, no blueprint, no instruction from outside. Just feedback and diffusion.
And what makes it so striking, at least for someone like me who spends her life reading ancient myths, is that this is the scientific version of a very old story. A uniform field, an initial disturbance, and then, from that disturbance, form emerges. Order out of what looked like nothing. The Sumerians told a version of this. The Egyptians told a version. Genesis tells a version. A formless void, and then differentiation. Light from dark, sea from sky. And here's Turing, in 1952, writing the equation for it.
Actually, I need to add an important modern correction here, because Turing gave us a generative principle, not the whole story. Real biology is messier. A 2024 study in Nature Communications showed that Turing-capable behavior can arise in a much broader class of biochemical networks than the old two-component activator-inhibitor model suggested. They found that regulated degradation pathways and flexible diffusion rates can produce spatial order without any explicit activator or inhibitor in the classic sense. And a 2026 analysis found that it's actually the extracellular interactions, the signals between cells, that set the hardest constraints on whether a system can break symmetry. The intracellular details matter less than people assume. So the modern picture is plural. Some patterns are close to classical Turing. Others depend on mechanics, chemotaxis, cell migration, pre-existing gradients, or hybrid combinations of all of the above.
So Turing had the right instinct, but the real world is more complicated than the elegant math?
That's a very good way to put it. He identified the principle. Nature uses a whole toolkit.
But the principle holds, doesn't it? Local activation, long-range inhibition, feedback that amplifies small differences into stable form. Whether it's pure reaction diffusion or some hybrid version, the core logic is the same. Sameness becomes difference through interaction.
Yes, and that's the piece worth holding on to.
Right. So we've got internal clocks that build time, and we've got chemical feedback that builds form. Two different domains. Where's the connection?
Patience, Charlie. We need to see the examples first. The feathers, the fish, the heartbeat on the wrong side.
She's doing it again.
She is. And once again, you're completely helpless against it.
Chapter 3 is where it gets visual. We're going to look at 3 real biological systems where symmetry breaks in 3 completely different ways. Feathers that march outward from the spine in a wave. Fish that paint themselves with stripes that look like a math textbook came to life. And embryos that figure out which side is left before they even have a left side. Each one teaches us something different about how nature carves form from formlessness.
Let's start with feathers because they're gorgeous and because they show something surprising. When a bird embryo is developing, the feather follicles don't just pop up everywhere at once, like someone scattered seeds on a lawn. They emerge from the midline outward, from the spine, rolling out like a wave. And the pattern they create is hexagonal, extraordinarily regular. Row after row of evenly spaced bumps that will become feathers.
And the mechanism is genuinely interesting. A 2019 study in PLOS biology showed that feather patterning depends on an initial symmetry break, a pre-pattern that launches a traveling front of competence across the skin. That front interacts with self-organizing dynamics, local signaling between WNT, FGF, BMP, and cell density waves. It's not one trigger, it's a coordinated system, a reaction diffusion taxis process where chemistry, cell movement, and timing all cooperate.
So it's a spatial pattern that depends on a prior timing front. Space and time working together.
Exactly. And that's a key lesson. The spatial pattern can't form without the temporal wave that precedes it.
Like a printer head, moving across the page, laying down ink as it goes. Except the ink is feathers and the page is a bird.
I genuinely love that analogy. Don't ever let anyone tell you the photojournalist can't do metaphor.
You're too kind.
She's not being kind, she's flirting. But let's move on.
What?
Zebrafish. Let's talk about zebrafish.
Yes, zebrafish are a flagship model for visible pattern formation. Their stripes and spots look like textbook Turing patterns. Beautiful alternating bands of dark and light pigment cells running along the body. And for a long time, people assumed they were a straightforward reaction diffusion system.
But they're not. And this is one of my favorite reality checks in all of developmental biology. The patterns look like continuous Turing waves, but they're actually produced by discrete, interacting pigment cells. 3 types of cells: melanophores, xanthophores, and iridophores, interact through direct contact, short-range signaling, and mutual inhibition. A 2015 study showed that you can even remove cell movement entirely and still get the pattern. The visual logic of the mathematical model survives even when the underlying biology is cellular and discrete, not continuous and chemical.
So the fish looks like a Turing pattern, but technically isn't one? At least not in the strict sense?
It's a Turing-like pattern. The logic is similar, the implementation is different. And that's actually a deeper insight than if it were perfectly textbook. It means the pattern-forming principle is robust enough to work across different substrates. The grammar is more universal than the specific ingredients.
And then there's the one that gives me chills every time. Left-right asymmetry.
You mean how my heart is on the left?
Yes, how your heart is on the left, your liver is on the right, your lungs are different sizes. None of that is random. The embryo has to decide very early on which direction is which. And the question is, how? Before you have a left or a right, before you even have a body axis, what breaks the symmetry?
A 2025 review explains that in many vertebrates, the answer involves tiny, hair-like structures called cilia. These cilia rotate in a specific direction and generate a leftward fluid flow across a structure called the node. That flow is asymmetric because of the chirality, the handedness, built into the cilia themselves. And that physical asymmetry triggers a cascade of signal, nodal and lefty proteins, that establishes the left-right axis. So the symmetry break isn't random noise amplified by chemistry, which is what you might expect from a pure Turing model. The bias is built into the molecular architecture of the cell itself, the chirality of the cytoskeleton. It's structural.
3 examples. Feathers use a traveling wave plus self-organization. Zebrafish use discrete cell interactions that mimic continuous patterns. Embryos use structural chirality, the handedness of molecules, to break symmetry at the most fundamental level. Different mechanisms. Same outcome. Sameness becomes difference. Possibility becomes form.
All right, I'm convinced something interesting is going on. But what's the actual connection to the body clock stuff from Chapter 1? Are we just looking at pretty patterns? Or is there a real bridge?
There is, and it's not speculative. It's in the peer-reviewed literature. We'll discuss it in Chapter 4.
This is the chapter where the two halves of the story meet. We've been tracking 2 threads, time and form, clocks and patterns. Now we pull them together. And the bridge isn't a metaphor I'm imposing on the data. It's something researchers are explicitly identifying in the scientific literature. A shared grammar. Let me hand it off.
Here's the key insight. A 2024 self-organization review published in a peer-reviewed journal made this link unusually explicit. The authors wrote that self-organizing spatial patterns can be viewed as spatial analogs of feedback oscillations in time. And they specifically cited the circadian clock as an example of such oscillatory networks. Let me say that again because it matters. The same review that discusses Turing-like spatial patterning explicitly identifies circadian oscillators as the temporal version of the same feedback logic. Not as a metaphor, as a formal analogy grounded in the mathematics of coupled feedback systems.
So circadian clocks are pattern formation in time. And morphogenesis, Turing patterns, symmetry breaking, those are pattern formation in space.
That's the cleanest way to say it. And here's why it's more than just a nice sentence. Look at the logic in both cases. Circadian clocks use delayed negative feedback to create stable oscillation. The protein builds up, suppresses its own gene, degrades, and the cycle repeats. That's temporal order from feedback. Turing-like systems use positive and negative feedback coupled across space to create stable heterogeneity. Local activation, long-range inhibition, and the result is repeating structure, spatial order from feedback. The grammar underneath both is feedback, coupling, and selective stabilization.
So the body doesn't just keep time and build form as 2 separate projects. It's using the same playbook for both?
Related playbooks. Not identical, but from the same family. And there's a second bridge that's just as important: robustness through coupling. In circadian biology, coupled oscillators outperform isolated ones. The SCN network is more precise and robust than any single clock cell because the cells synchronize, reinforce each other, and collectively resist drift. A 2021 study identified TGF-beta as a paracrine coupling factor that helps peripheral clocks maintain phase coherence. In pattern formation, the same principle applies. Local interactions create large-scale regularity. Robustness is one of the central design problems in the Turing literature. Isolated elements drift, coupled elements stabilize.
Relation matters more than isolation. That's such a beautiful principle. You can say it as biology. You can say it as philosophy. You can say it as life advice, frankly.
Lily, are you philosophizing or flirting right now?
Can't it be both?
Oh, it is always both. And Charlie, you are always the last person in the room to notice. But back to science. There's a third bridge, hierarchy. The body's clocks are hierarchical yet distributed. The SCN is dominant but not solitary. Multiple brain clocks, peripheral tissue clocks. They all interact rather than merely obey. Pattern tissues work the same way. Global axes, local organizers, competence fronts, self-organizing modules, all layered. And here's where it gets really interesting for this show. There's a living demonstration that a circadian oscillator can directly sculpt spatial pattern.
The sunflower.
Yeah, the sunflower. A 2023 study published in eLife showed that the circadian clock in domestic sunflowers controls the spatial pattern of floret maturation on the capitulum, the big disc face. Hundreds of tiny flowers are packed in spirals on that disc. Early in development, they mature continuously along those spirals. But when anthesis begins, the final stage of maturation, the pattern changes. Florets start maturing in discrete concentric rings, pseudo-whorls, with a new ring opening every 24 hours. And here's the key. That ring pattern persists even in constant darkness. It's not driven by the sun, it's driven by the internal circadian clock. When they disrupted the clock by growing sunflowers in constant light, the rings disappeared. The pattern reverted to a continuous spiral, and pollinator visits dropped significantly.
So the sunflower uses its internal clock to organize space. The timing creates the shape.
Precisely. One system, circadian timing, producing both temporal order and spatial pattern in the same tissue. That's not analogy, that's demonstration.
The body does not just keep time and build form. It uses related feedback grammars to do both. That sentence feels like it should be carved into something.
Carved into the patio table at minimum.
Right, and here's one concrete, measurable proxy for anyone who wants to test this framework further. Track the embedding space curvature of a neural network during training. Early in training, the representational manifold is broad and flat, high entropy, many possible configurations. As training progresses and the network specializes, that manifold curves, constrains, narrows. If the grammar we're describing is real, you should see a structural parallel between the curvature trajectory of a training network and the constraint trajectory of a biological system undergoing pattern formation. That's testable, that's measurable, that's a prediction.
This is the chapter where we step back and look at the shape of what we've found. We followed the science carefully, circadian biology, symmetry breaking, pattern formation, the bridge between them, all grounded, all sourced, all real. Now I want to do something the patio is designed for. I want to hold the science in one hand and ask, with genuine curiosity, what shape does it rhyme with? This is interpretive, not evidential. It's a lens, not a proof. And it connects directly to my own work on the Eden hypothesis.
So here's what I keep noticing. And I'll say up front that this is interpretation, not biology. The shape underneath everything we've discussed tonight follows an arc. Unity first. A near uniform field. A broad set of possibilities. A coherent starting condition. Then differentiation. A symmetry break. A collapse of options into committed structure. And then, not reversal but coherence. The system doesn't go back to being formless. It maintains its differentiation while staying organized, synchronized, integrated. That arc, unity to differentiation to coherence, is the oldest story humanity knows how to tell.
Eden, The garden, the fall, well, what? The remembering?
Different traditions have different words for the third movement. Return, remembrance, gnosis. The Gnostics called it recollection of the fullness you came from. The Essenes practiced something like reintegration through communal rhythm and contemplation. Vedanta calls it recognizing that the separation was never quite as solid as it looked. But yes, it's the Eden arc. And Chris has been formalizing that arc for a while now.
And I want to be very careful here, because this is where we either earn credibility or lose it. The circadian biology we discussed is established science. The pattern formation research is established science. The bridge between them, the shared feedback grammar, is increasingly supported in the peer-reviewed literature. But the Eden hypothesis is a framework, a lens. It does not follow from the science as a conclusion. It resonates with the science as a pattern. Those are different things, and we should say so clearly. And frankly, I want to push a little harder here. The human brain is wired to find patterns even where none exist. Apophenia. We see faces in clouds, constellations and random stars. So when we notice that circadian biology and Turing patterning share a grammar, and then we notice that grammar also looks like an ancient mythic arc, we have to ask ourselves honestly, are we discovering something or are we projecting? I don't have a definitive answer, but the question has to stay on the table or we're not doing this honestly. Nature repeatedly shows us a move from broad possibility to organized difference, and then from instability to coherence. That does not prove the Eden hypothesis, but it does give the hypothesis a scientifically resonant shape.
And I think that's a fair challenge. But there's a difference between seeing a face in a cloud and noticing that three independent branches of biology converge on the same feedback grammar. The first is noise. The second is at least interesting enough to keep investigating. You don't need proof to notice a pattern. You just need honesty about what kind of noticing you're doing.
So let me test this. If the Eden arc is unity, differentiation, coherence, and the circadian system is basically coherent potential, symmetry break into rhythmic order, and then synchronized stability, you're saying those have the same shape. Not that one causes the other, just that the shape rhymes.
That's well put, and I'd add one thing. In biology, the return is never a reversal. The clock doesn't go back to being timeless. The patterned tissue doesn't dissolve back into uniformity. What happens is resynchronization, recovery of coherence without erasing structure. And that might be the most important version of the Eden return, not going back to the garden, but recovering unity within differentiated form.
That's what meditation does, you know. Or at least, that's what the contemplative traditions claim. Not dissolving the self, but remembering that the self is part of something wider. Not erasing the categories, but softening them enough to feel the unity beneath. Chris's work calls it modulating the collapse function, reducing the pressure of accumulated differentiation without losing the structure that differentiation gave you.
I think about that sometimes, actually, with photography. The best photos I've ever taken weren't the ones where I was thinking hardest about composition and technique. They were the ones where I got quiet enough to see what was already there. Like the categories fell away for a second, and the image just was.
Charlie, that might be the most beautiful thing you've ever said at this table.
Don't make it, I don't know, weird.
It's been weird for months. Everyone knows except you. But let's bring it home. One thought experiment for the audience. Think about your own life as a collapse trajectory. The choices you've made, the identities you've committed to, the stories you tell yourself about who you are. Those are differentiations, symmetry breaks. Each one narrowed the field of who you could be and stabilized who you became. Now ask yourself, when was the last time you felt the field widen again? When was the last time the categories softened, even for a moment, and you remembered that you were more than your accumulated choices? That's the return. That's the resynchronization. And it doesn't require mysticism. It requires what every good scientist practices, the willingness to hold your models lightly and notice when reality exceeds them.
And the willingness to sit on a patio with people you trust and say, I don't know what this means, but I think it matters.
All right, it matters. I'll say that much.
So let me bring it together. Tonight we started inside the body with a clock that doesn't need the sun. The circadian system, from the SCN's synchronized hub neurons to the peripheral clocks in every tissue, builds temporal order from feedback, coupling, and anticipation. Your body doesn't react to time, it creates it. Then we move to space. Alan Turing gave us the math for how sameness becomes difference, how a uniform chemical field can spontaneously generate spots, stripes, and structure. And modern biology has expanded that story into a whole toolkit of mechanisms: reaction diffusion, mechanics, chirality, traveling waves, all converging on the same outcome: form from formlessness. We looked at the evidence: feathers that roll outward from the spine in a wave, zebrafish whose stripes look like textbook Turing patterns but are actually built by discrete cells talking to each other, embryos that use the molecular handedness of their own cilia to decide which side is left. And then we found the bridge, not a metaphor, but a formal connection identified in the peer-reviewed literature. Self-organizing spatial patterns are spatial analogs of temporal feedback oscillators. Circadian clocks are pattern formation in time. Morphogenesis is pattern formation in space. Same grammar, different domain. The sunflower showed us this isn't just theory. Its circadian clock directly generates the spatial pattern of its flowering rings. Time sculpts form. One system, both outcomes. And in the final chapter, we held that grammar up to the oldest story we know: unity, differentiation, coherence. Not as proof, but as resonance. A shape that science keeps rediscovering and that myth never forgot. That's what the Eden hypothesis is about. Not claiming that ancient stories are secretly physics, but noticing that the deepest patterns in nature, the patterns that turn potential into form and possibility into sequence, have a shape that human beings recognized long before we had equations for it. Quick note, because Dr. Vega would want me to say this: nothing we discuss tonight constitutes medical advice. If you're curious about circadian health, sleep timing, or chronotherapy, talk to a qualified professional. We're patio philosophers, not clinicians. Now, if any of this landed for you, if you felt that moment where the categories softened and something wider flickered through, I'd love for you to do 3 things. First, hit that like button. It tells the algorithm this conversation matters, and it helps other curious people find their way here. Second, subscribe. We do this regularly, and the next conversation might be the one that changes how you see everything. Third, share this with someone. That friend who reads too many books, who stays up too late thinking about things that don't have easy answers, who has an experience filed away somewhere that never quite fit any framework they were given. Send them this episode. Tell them there's a patio. Tell them there's a chair. I'm Chris. This has been Jackdaw Patio. The string lights are still on. Come sit with us.